What did I do? Hi, can you guys see and hear me? This is the continuation of my notes on cell and biology and life science in a nutshell. We're reviewing and studying. We have reached the section of DNA structure, and earlier we learned about DNA, cell division. So now we're learning about the structure of the genes, um, genetic material, which are DNA. So, DNA recombination, which is unique to meiosis. In meiosis, you have which occurs in gametes or sexual cells, which are for the purpose of passing on genetic material to a newly produced organism. So each parent cell contributes one strand of DNA, a haploid, or half the amount of genetic material. And once these two strands and the two strands from each parent combine and form a complete set of DNA in the offspring. However, the important thing about meiosis and sexual reproduction is that genetic recombination occurs of the DNA molecules from the parents. Um, so you have a multiple a number of combinations of genes you can have, and this is why uh, siblings don't look identical. There's differences in their genes based on how the recombination occurred in the parents' chromosomes in the phase of DNA synthesis and in the prophase stage of meiosis, where the genes are being synthesized and they recombine. Initially, when you have DNA synthesis, one a strand of DNA doubles, the amount of DNA doubles. So, two strands. You have two strands that double, and then once they double, they divide again, and then they divide a second time. So, from the original double strand, you get um, you get first you get four strands, which are tetrad, and this is when you have crossover occurring between the four strands, and then the four strands divide into two, and then the two divide into one. So this one strand from the one parent, so one from the mother and one from the father, combine to form the new offspring cell or organism called a zygote, which has two strands again, which is the required amount of genes to start out. And then once you have that, you can just have regular mitosis in order to multiply the cells, and development can continue. So the holiday junction is an intermediate genetic recombination involving four separate DNA strands. Recombination allows chromosomes to exchange the genetic information and is important in evolution of new proteins as well. Also, DNA repair responds to breaks, etc. The recombination reaction is catalyzed by recombinases, and the enzyme proteins. The first step in recombination is a double-stranded break caused by either an endonuclease or damage to the DNA. Along the pair of chromosomes, swapping occurs of one strand for another. The recombination reaction is halted by cleavage of the junction and religation of the released DNA. So that's how that occurs in DNA. So in summary, eukaryotic nuclear DNA is located within chromosomes. Human genome has approximately 3 billion base pairs of DNA arranged into 46 chromosomes. So 46 is the diploid number. So essentially 23 homologous chromosomes, which means they're in pairs. And the pairs, or the homologs, are determined by similar chromosomes pairing with one another. So 23 pairs equals 46 is the diploid or full number of chromosomes in the human. Protein synthesis is when genes in the nucleus are used to produce proteins. This occurs on the ribosomes. The protein that DNA codes for determine the characteristics and the processes of the organism. In protein synthesis, a succession of tRNA molecules are charged with the appropriate amino acids, which are brought together with the mRNA molecule and matched up by base pairing through the anticodons of the tRNA with successive codons of the mRNA. The amino acids are linked together to extend the growing protein chains. The tRNAs no longer carrying amino acids are released. The whole complex of the process is carried out by the ribosome. So this is called translation. So the ribosome is made of rRNA, or ribosomal RNA, which and also 50 different proteins, which may help with the translation process. So 2 main chains of RNA, or rRNA, and proteins as well. The ribosome latches on to the end of an mRNA molecule, moving along it, capturing loaded tRNA molecules, joining together their amino acids to form a new protein chain. So this picture is showing this process. And note RNA is transcribed in the nucleus. Once RNA is transcribed, it is transported to the cytoplasm, translated by the ribosome. Okay, so translation is the assembly of amino acids by the ribosome, which is an essential part of biosynthesis, aka synthesizing proteins. So you have transcription is prior to translation, which is the RNA being synthesized from DNA template. So first you have transcription, and then subsequently, once you have RNA made in the nucleus, you have translation in the ribosomes, which is amino acids assembling off the RNA template, which serves as an intermediate for synthesis of polypeptide chains. So here you see the picture of the DNA, the mRNA, transcription of the DNA, then the RNA leaving the nucleus, transported to the cytoplasm, and it's synthesized into proteins using tRNA and helper protein molecules to form an amino acid chain, or aka a protein. So that's the process. Thank you guys.